Good morning, everyone. I'm Jinju. First of all, I want to thank our department and our seminar director, Dr. Travers, for giving me this wonderful chance to share my work. So as shown here, so today I'm going to talk about exosome-mediated effects of exercise on vascular disease. So vascular disease is a general term for any conditions that affect the network of blood vessels. If this injury happens in the brain vessels, it might cause ischemic stroke or hemorrhagic stroke. If the injury occurs in the coronary artery, it might cause myocardial infarction. So if the injury occurs in the aorta area, it might cause aneurysm. In the legs, the limbs like legs, it will cause peripheral arterial disease. So the exact cause of vascular disease is unknown, but there are many risk factors like physical inactivity, obesity, diabetes, hypertension, hyperlipidemia. All of these will contribute to the progress of vascular disease. Everybody knows exercise is good for our health. Here is just an overview of the benefits, effects of exercise on cardiovascular system. So in the blood system, exercise can change the lipid profile. It can improve insulin sensitivity. Somehow exercise can modulate the circulating progenitor cells in the blood. So from the heart, it can change the mitochondrial biogenesis, but eventually the goal is to increase the cardiac output, improve the heart infection. So blood vessel system is also is my focus. So in this system, exercise can increase endothelial nitric oxide synthesis expression and increase the nitric oxide bioavailability. So two of these are very important for endothelial function. So, but although we have seen the robust effects of exercise, but the exact mechanism remains unknown. Endothelial progenitor cells, we call the EPCs. They are bone marrow-derived immature cells. They can migrate to injury area and differentiate into endothelial cells for endothelial repair. There are some studies show exercise can modulate EPCs. So in this study, they used myocardial infarction mouse. So these mouse did 2 weeks moderate exercise, and they found the EPC function is improved. So this work was done in human studies. So either healthy individuals or chronic heart disease patients, they do one-time exercise can increase the release of EPCs from the marrow. So this suggests that exercise can modulate the EPC function and the level. EPCs play an important role in the vascular disease, but in some disease conditions like hypertension and diabetes, their level is decreased and their function is compromised. Our lab a few years ago, demonstrated that EPC infusion can protect brain for acute ischemic injury. So the potential mechanism is through cerebral blood vessel protection and promote angiogenesis. So later after that, when I was in Dr. Chen's lab, I was doing my PhD project. At that time, we found EPC released extracellular vesicles. They also exhibit protector effects on endothelial cells, we call the ECs, against the hypoxic injury. So exosomes, I know some labs, like Dr. Travis' lab, Dr. Sahu's lab in our department, they are also doing some work on microparticles. So exosomes is a major type of extracellular vesicles. They can be released by nearly all types of cells. They present in the body fluid circulating in our body system. So as shown here, exosomes are bilayered. On their membrane, there are tetraspan proteins. These proteins have been used as markers for exosome. Exosomes carry the RNAs, DNAs, proteins, and the metabolites of their parent cells. Previously, exosomes, these vesicles, like include vesicle exosomes, they are thought to be just like a garbage bag to dispose the garbages of parent cells. But nowadays, it's widely accepted that these exosomes play important role in cell-cell communication. So as shown here, these exosomes are released from donor cells. So once they release, they could be uptaken by recipient cells. In these recipient cells, they will release their cargoes, like the microRNAs, messenger RNA, lipids, or proteins. So these cargoes presumably to change the function of recipient cells. So exosomes not only affect other cells nearby, they can also talk with the cells in distance because they can circulate in our system. So here I want to emphasize the role of microRNAs in exosomes. So the contents of exosomes are rich with microRNAs, although the sorting mechanism we don't know. There are a couple of studies show that if we lock out the microRNAs in their parent cells, the cells release the exosomes function will be changed. So this study, they lock out mesenchymal stem cells, DROSA. So DROSA is a nucleus involved in microRNA production. If they lock out DROSA, so they generate microRNA deficient exosomes. So these exosomes lose their function in protected acute kidney disease. In this study, they lock out microRNAs in the target cells. So they found that these mesenchymal stem cells exosomes lose the ability to promote exosome outgrowth. So these studies shows that microRNA plays an important role in exosomes. So MIR-126 is an endothelial-specific microRNA. It can modulate angiogenesis and vascular integrity, but its circulatory level is decreased in ischemic stroke. On 2016, we found that EPC extracellular vesicles function is compromised in diabetes patient plasma. We found out that it's because of the reduction of their carried microRNA. I mean, they carried less microRNAs. There are another study published last year. They found the overexpressed MIR-126 in EPC exosomes can promote their function on thrombus resolution in the animal model. So this suggests MIR-126 plays an important role in the EPC-derived extracellular vesicles. Recently, exercise is a hot topic, and researchers try to see if after exercise, the extracellular vesicles, their release will change a lot because vesicles can release when cells are under stimulation. So exercise is a kind of stimulation. This study was performed on rats. So these rats performed acute exercise at a different intensity, and then researchers found that their level is increased. Another group From Australia, they recruited healthy volunteers, did a one-time exhaustive cycling exercise. They collected the blood before the exercise, immediately after exercise, and 4 hours after exercise, and they found that this one-time exhaustive exercise can increase the release of EVs, extracellular vesicles, but it back to normal level after 4 hours. So in this study, meanwhile, they performed mass spectra to analyze the protein contents of these extracellular vesicles. They identified more than 300 types of proteins released in these extracellular vesicles. They did not perform function study on these exercise. Besides, there's no study show the origin of these. Which types of exosomes are these exosomes from? I mean, it's a pool of vesicles, but which type of exosomes is most affected by exercise remains unknown. But till now, we know exercise can promote the release, modulate the release of exosomes. So what we are interested in is if these exosomes can modulate the function of exercise or vascular disease. Because as I mentioned, EPCs and MIR-126 are important for vascular disease. So we are wondering whether exercise can modulate the release and these MIR-126 level in like EPC-derived exosomes. So with these ideas, we hypothesized that exercise has beneficial effects on ischemic stroke through modulated circulated EPC exosomes, which can affect the function of endothelial cells and neurons by their carrier MIR-126. So to test our hypothesis, so first we tried to determine different intensity exercise effect on the release of these circulating EPC exosomes and their carried MIR-126 level. Further, to detect these exercise-derived exosome function, we used a cell injury model with endothelial cells and neuron cells. Further, we tested these correlations in the animal model. So this is the major ideas of my postdoc grant. Here is the general timeline of my study. So we used C50 mice exercise for 4 weeks at 2 different speeds. So each time is 1 hour, and they did exercise like 5 days a week. After exercise, some cells were sacrificed for blood and brain tissue collection, and some mice were subject to MCO surgery to induce ischemic stroke. So after we observed 2 days or 28 days for acute injury or chronic recovery. So here shows the exercise we isolated from the circulating plasma. So these curves are 3 types of EPC exosomes. So as shown here, we can see the size distribution was not changed, but their number, the number of these exosomes were significantly increased, and this is intensity dependent. We confirmed the exosomes origination by using exosome markers like CD63 and TSG101 and EPC-specific markers. So after I isolated these exosomes, I want to test if there's any function difference. So we injured in vitro cell model. We used the endothelial cells here. to mimic the ischemic injury in diabetic condition. So we treat these cells with high glucose and hypoxia. These cells displayed, a higher portion of cells displayed apoptosis and angiogenic dysfunction. We evaluated the tubule formation and the migration ability of these cells. So after I induced the cell model, we did a co-culture assay, and we found that so after 24 hours co-culture, the 3 types of exosomes can rescue partial of endothelial cells from high glucose hypoxia-induced injury. But as shown here, so we can see exercise mouse-derived exosomes had better effects, especially this one had the best effect. These are from moderate exercise mouse. So for this endothelial function analysis, we found these 3 types of exosomes increased the migration and tubule formation ability. Similarly, the exercise-derived exosomes had better effects, and the moderate exercise mouse-derived exosome had the best effect. So we are wondering, because during the co-culture we used a similar amount of exosomes, the same amount of exosomes. So why there is effect difference? So we try to think about this. So we are wondering if miR-26 is there a difference of miR-26 level in these exosomes. So we extracted the RNA and performed the qRT-PCR, and we found that yes, after exercise, no matter 5 or 10, the relative expression level of this miR-126 is increased, and a much higher increase in these moderate exercise mice. I performed that on wild-type mice. We also confirmed this in a diabetic mouse. So we used diabetic mouse, did exercise, and then isolated the cells, evaluated the miR-126, and we found that these 2 types of exercise do improved the increased the miR-126 level. So further, so we want to see if this effect difference is caused by miR-126. So we think about we can lock down these miR-126 in the target cells, because these exosomes are from mouse, so we cannot modify it, but we can do this work on the target cells. So, back to these target cells, the endothelial cells, I measured the miR-126 level after co-culture. So I found the 3 types of exosomes do increase miR-126 level. The best effect is here. So as I mentioned, miR-126 is endothelial specific. That's why we observe an increase here. So here, this suggested that EPC exosomes can transfer these miR-126 into the endothelial cells. So let's lock down. So we use the miR-126 inhibitors on these endothelial cells before co-culture. So we found that after we locked down miR-126, so the effect of EPC exosomes on the apoptosis in their angiogenesis function is blocked. So from this data, we can conclude that exercise can increase this function, the EPC function on rescue endothelial cells against hypoxia or high glucose injury is through conveying miR-126. So for the downstream, we analyzed the SPREAD1. SPREAD1 is a potential target for MIR6. So in these target cells, endothelial cells, we found the SPREAD1 expression was decreased, especially in this group, the exercise from moderate exercise. But if we lock down MIR126, SPREAD1 expression is back to as compared to its control group. So VEGF has been previously mentioned to be electrically regulated by SPREAD1. So in the targeted cells, we measured the VEGF expression, and we found in the treatment groups, VEGF expression was negatively correlated with the SPREAD1. So from here, so we know, we can conclude that exosomes can convey MIR126 to the targeted cells, endothelial cells, and then these MIR126 can block SPREAD1 expression and further increase VEGF expression, then reach the therapeutic effects. As we know, neurons are important brain cells. So we also measured we also evaluated the function of these exercise-derived EPC exosomes on these neuron cells. So we the neuron cells subjected to hypoxia injury. So as shown here, after treatment, exercise-derived exosomes do improve the viability and apoptosis. It also restored the neurite growth after co-culture. So for in the neuron cells, we measured for mechanisms, we used PR3K, because PR3K active pathway is a classical pathway for cell survival. So we blocked PR3K pathway using classical PR3K inhibit, and we found the beneficial effects were significantly decreased. And here shows AKT expression, the phosphorylation of AKT. And so after we blocked MIR126, lockdown MIR126, we found phosphorylation is decreased. So far, I evaluated the EPC exosome function on the two types cells in vivo. So in the virtual animal model, we, as I mentioned, after exercise, some mice was subject to MCL injury, MCL-induced ischemic stroke. So we we sacrificed the mouse and collected the brain tissue for TDC staining, as shown here. So the red color stains live cells, the white part shows infected area. So we summarized data. We found that exercise do increase infected volume. So on 28 days, we performed a behavior study. So here I did a color test and an enhanced remove test. So color test is like this. We have two paperboard, make a 30-degree angle, put the mouse in the middle of this space and observe if they will turn left or turn right. So in normal mouse, if without disease, there is an equal chance to turn left or right side. But after stroke, because one side has disability, so they cannot turn. So we will observe the difference. So as shown here, day -1 means before surgery, day 0 means after surgery, the same day after surgery. So we see before surgery, equal chance, pretty much equal chance both sides. So after surgery, 90% they will turn left because the other side has a disability, as I mentioned. So after 28 days recovery, these exercise mouse had better recovered, as shown here. So they get more, there's a higher chance for them a lower chance for them turn to left, I'm sorry. Yeah. So another test we did is the adhesive remove test. So this one is we put the tape, the adhesion tape into the probe, and then we observe how long for the mouse to take off the tape. So the longer, the worse. So as shown here, on day one, just take a couple of seconds, they can quickly take it off. But after surgery, it takes almost 40 to 43 seconds to take off. This means surgery is successful at least. So after 28 days recovery, we find in the low intensity exercise mouse, they take short time to remove the tape, and even shorter in these moderate exercise. So this means exercise do maybe have a prevent effects for reduce the ischemia injury. So to summarize this part, so we have shown that exercise can increase the level of CEPC exosomes and their MIR-126 cargo in both types of mouse. And we found the moderate exercise had better effects than no exercise in our study setting. We showed that exercise can regulate the protective effects of these exercise-derived exosomes on the two types of brain cells. The potential mechanism is MIR-126. So in the animal model, we demonstrated that MCO-induced ischemic injury is reduced in both types of exercise, but the moderate exercise had better effects. So these suggested that exercise has preventive effects on ischemic stroke. For this project, I will continue to do more work on evaluate the exercise effect on ischemic stroke in acute phase and chronic phase, and then I will do analysis of the correlation of the EPC numbers and MIR-126 level in exosomes with these outcome parameters of ischemic stroke. So hopefully I have convinced you that exercise can moderate the function, moderate the level of MIR-126 in exosomes and can change the function of these exosomes. We know adipose tissue in diabetic condition, adipose tissue inflammation is a big concern. It participates in the pathology of diabetes vascular dysfunction. So I'm interested in whether exercise could have beneficial effects on these diabetic vascular inflammation through modulating adipose tissue derived exosomes. Especially I will focus on PVAT. So PVAT are perivascular adipose tissue. So first I will give a brief introduction of adipose tissue. We know it's the largest endocrine organ. It disperses throughout the body, range from 5 to 60% of the body weight. There are two types, adipose tissue, white or brown. So these adipose tissue can secrete active substance to regulate inflammation, metabolism, or thermogenesis. So PVAT is a special adipose tissue. They surround almost all the blood vessels except the vessels in the brain. I mean, there's no PVAT tissue in the brain brain vessels. Previously, they are thought to provide mechanical support on these vessels. But nowadays, more and more studies show that they participate in vascular dysfunction. These PVAT can be classified either brown or white, depends on the location. So in physiological condition, So the PVAT surround the thoracic aorta more close to brown adipose tissue. But for like these mesenteric PVAT, their properties are more close to white adipose tissue. But this might change in disease condition. As I mentioned, some studies have demonstrated PVAT participate in the development of vascular disease. This group is from Kentucky, so they found that in angiotensin II infused obese mouse, the increased PVAT inflammation surrounding the abdominal aorta is associated with a higher ratio of aneurysm in the abdominal aorta. Another group also showed that the immune cells like macrophages, T cells, B cells, these are involved in aneurysm formation, have been observed in PVAT. So this also suggests that PVAT inflammation is involved in vascular disease. This group is from UK. They used ApoE lockout mouse. They found that PVAT inflammation can precede plaque formation and promote the development of endothelial dysfunction in these mice. But so far, to my knowledge, there is no study on PVAT exosomes in the vascular disease. They do have some studies to show the exercise can reduce adipose tissue inflammation. Most of these studies was done in the white adipose tissue. So as shown here, this group, they used high-fat diet feed mouse. They found exercise can decrease cytokine and some adhesion molecules release in the gondral adipose tissue, which is a white type adipose tissue. Meanwhile, they found the macrophage, type 2 macrophages expression is increased in those locations after exercise. Later on, they found that exercise can prevent macrophage infiltration into the adipose tissue. So this shows exercise can decrease adipose tissue inflammation, but so far no studies show exercise effect on PVAT inflammation. That's why I'm going to do some work in this area. So here we did some preliminary studies. So first thing we want to confirm is exercise can decrease adipose tissue inflammation in diabetic mouse. So we let the mouse run at a moderate speed, and then we collected the stromal cells from the adipose tissue, and we used macrophage marker F480 to analyze the macrophage infiltration in the adipose tissue. We found that after exercise, the percentage of macrophages was decreased. So we found a decreased macrophage infiltration. We want to see if the types, the polarization of these macrophages changed a lot. So we further used CD86 and CD206 to analyze the two subpopulations of macrophages. As shown here, so the M2 in diabetes condition, type 1 macrophages are more than type 2, but after exercise, we saw more type 2 macrophages in the adipose tissue. I forgot to mention, macrophage 1, type 1 macrophage is they can release pro-inflammatory cytokines. By type 2 macrophages, they can release anti-inflammatory cytokines. So from here, so we generally conclude that exercise in DB mice can increase the macrophage polarization. So we also did an immunofluorescence study on this adipose tissue. Consistently in diabetic condition before any intervention, we can see more M1 cells present in the adipose tissue, but after exercise, so more M2 macrophages. So as shown in the merger, the yellow color, because we used 2 different antibodies to identify these cells. So from here, we can conclude exercise can decrease adipose tissue inflammation in the white adipose tissue. So further, we evaluated whether the PVAT inflammation can be changed. So here we collected the PVAT from the altar, along the altar. And then in DB condition, we found more M1 cells present in these tissues. But after exercise, we saw more M2 types cells present in these PVAT tissue. So that means exercise can decrease PVAT inflammation. But there are lots of open questions. We don't know how this is happening. What the mechanism, if whether PVAT exons were involved or there are other cells-derived exons or cells directly involved, we don't know. Besides the inflammation evaluation, we also measured the ultra remodeling and the collagen deposition, because collagen deposition is also an important marker for inflammation in the blood vessel. As shown here, we performed the HE staining in DBC condition, in diabetic condition, the wild type mouse. As compared to this wild type mouse, we see DB mouse has a thick intimal layer, which is the medial layer of the blood vessel. But after exercise, this kind of remodeling was reduced. For collagen staining, we performed the Mason staining, as shown here, this blue. So we measured the blue area, and as compared to control mouse, diabetic mouse has higher level of collagen deposition. This is consistent with the previous study showing more inflammation in the adipose tissue area. So after exercise, the collagen deposition was decreased. But again, similarly, we don't know this remodeling change, collagen deposition change is related to PVAT or not, or other cells, other immune cells, we don't know. But we can confirm is that exercise can decrease the vascular remodeling, and further it might improve the function of these vessels, like the aorta. So after we observed that, so we are wondering whether these exons might have an effect on the blood vessel or not. So we used smooth muscle cell. So we induced a smooth muscle cell injury model with high glucose, and then we co-culture these injured cells with the PVAT exons from the sedentary mouse in the exercise mouse, and we found that So these exosomes are from diabetic mouse without exercise. They had detrimental effects on smooth mother cell proliferation, and they promote cytokines and like monocyte MCP1 release. But after exercise, we see the co-culture with these exosomes from exercised mouse decreased smooth mother cell proliferation and their cytokine release. So this showed us exercise can change, alter at least the function of PVAT exosomes. But how they did that, this is because of their carried microRNAs or proteins or other components. We don't know. And another question is how exercise change the release of exosomes. We don't know either. But with this preliminary data, so in the future, so I'm going to illustrate whether these PVAT exosomes participate in the vascular inflammation remodeling in diabetic condition. So I want to determine whether this has pathological effects on the vascular function in diabetic condition, and whether we can do like after exercise, we can change the function of these exosomes. So the second one we are going to ask is whether these exosomes are mediators for exercise modulated vascular protection in diabetes. So these are open questions. Besides this, as I mentioned, there are lots of other open questions like the mechanisms, how exercise change the release of exosomes, what are the sorting mechanism, biogenesis mechanism. All of this I don't know, but in the future, I plan to work in this area. So with this, I would like to end my talk, and thanks to my group. So first, thanks to Dr. Biao for her mentor during the past years, and my PhD mentor, Dr. Chen, for his years support. And everybody in our lab, our master students, and Dr. Wenfeng Zhang, Shu Zhen, and our PhD students. Also, I want to thank all the faculty and staff in our department. Especially Dr. Kuo, I remember I did, when I was in PhD, I did a rotation in his lab in Dr. Xu's lab also. Yeah, and I want to thank Cherry and Dr. Ores. She recruited me when I joined the master program and later joined this area. So I want to thank my funding resource from AHA and Dr. Beals from ADA and the NIH. So with that, I will be happy to take any questions, and thank you for your attention.
First of all, what a wonderful talk. What very strong data. I think that reflects nicely upon your group. So my question has to do with the really intriguing data that you had indicating that moderate exercise will change the polarization of macrophage from an M1, a pro-inflammatory, to an M2, an anti-inflammatory, in the vascular, the PVAT, the fat around it. Now, when you did those experiments, did you also take other organs to see if this macrophage in blood, to see if this macrophage polarization was specific for that area, or was it a generalized phenomenon?
We collected blood. We collected the mesenteric arteries and the thyroid adipose tissue, but I cannot give you an answer at this moment because we are doing some work on that, yes. We don't know, we are not sure if this macrophage polarization is specific in this area or it could be in other areas, other PVATs, yeah.
Or in other systems, do you see differences in, for example, the spleen or real important organs like the skin?
Yes. Yes.
For the benefit of those of us who are not familiar with your microRNA, could you explain a little bit more about how it works?
Oh, so you mean how it works in the target cells?
What does it do to the cell that takes it up?
The cell takes it up. So usually, if it was the microRNAs were uptaken by the target cells, so they will either bind to the downstream target cells. So this is it's regular, it's normal function. So they will either stop the transcription of downstream proteins.
What does it alter transcription of?
How do they? That's a good question, but I'm not sure at this moment. For how the exosomes so you ask about microRNAs released by these vesicles and then uptaken by the target cells, how they make changes of target cells?
Well, what's going on? I don't understand.
It's what genes does it target?
So it depends on what type of cells they were uptaken. Yeah, so to my understanding, once they were uptaken by the target cells, they might bind to different downstream proteins, infect the transcription of these proteins.
Questions from the students? Of course, the students don't have time to exercise instead of working all the time. Yeah, your brains. The process, what do you think is doing the effects of exercise? Is it the release of endorphins, for example, or is it just the high blood pressure? Can you measure, can you mimic the effects of exercise by just increasing the blood pressure? For example, in a hypertensive mouse, could you repeat the same would a hypertensive mouse cause increased exosome release?
Actually, we did some work on the hypertension mouse. We let the hypertension, because we have the transgene mouse in our lab, so we let these mouse did an exercise, similar speed of these. We did exercise. We plan to collect the blood and do analysis of the PVAT exosomes, because the renin-angiotensin system is really important in the hypertension. So in these PVAT, they contain the components of the renin-angiotensin system. So we are wondering, after exercise, are there any change? If there is change, there's some potential. So that's why we have some hypertension mouse do the exercise running. Yeah.
So did the water destroy the example before the
Yes.
So how do you think it is possible after the stroke? So you're doing the exercise and die more?
After exercise, theoretically, they supposed to provide benefit. Promote recovery after surgery if they did a proper intensity exercise, because exercise can increase the increase the circulating endothelial progenitor cells in the circulation system. So these EPC cells can promote recovery in local area. And besides, as we show here, exercise can promote the release of EPC exosomes, EPCs exosomes, I'm sorry, yeah. So I think, yes, after surgery, if they do rehabilitation, it is supposed to have a positive effects on the recovery of stroke.
Yes. Do we think that there can also be differences with age? How old were maybe the mice were used, or the adult, or so it maybe in humans with differences in their age types, could we see the same effects?
That's a really good question. Age is a really important factor we should consider when we do either animal study or human study. I'm not sure about how, the changes of PVAT in different ages. But as we back to Dr. Travis' question, like we said, the renin-angiotensin system, like ACE2, they are decreased as age increase. When you get older, ACE2 level is decreased. Previous study shows PVAT contains ACE2, but theoretically they must decrease if they get older. But I don't have solid data to answer you, but yeah. Thank you.
As our tradition.
My pleasure. Thank you.